Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Freddie Lawton from Business Review, and I'll be your host. It is our pleasure to have Vera Nova with us today, who will be discussing unmatched viral particle analysis in vaccine and gene therapy development. Today's guest speakers are Dr. Josefina Nilsson, who is the Head of EM Services at Vera Nova, and Martin Reiner, who is the Manager of Strategic Development at Vera Nova. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link they receive via email to rejoin the session. In order to ask questions, you can send them in via the questions widget, so just type them into the box at the top left-hand corner of your screen and click on Submit. We allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. If you click on the survey widget, there are a few questions in there for you to answer, and you can do this during or immediately after the webinar. Also, if you click on the green resource list widget, you'll find a couple of relevant links from our speakers. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now please allow me to welcome Josefina.
Thank you very much, Freddie, for the introduction. So today, Martin and I will talk to you about viral particle analysis in vaccine and gene therapy development. However, this approach can of course also be used for other nanoparticles. So first, please let me give you a short introduction to Vera Nova. Vera Nova is a Swedish company located in Stockholm. We do offer unique services for nanoparticle characterization and virus clearance testing together with hardware and software for advanced electron microscopy analysis. To be able to do this, we have and need to have experts in interdisciplinary fields, like electron microscopy, of course, imaging analysis, programming, mathematics, virology, and structural biology. So by combining the expertise that we have with our in-house developed software, VAS, we offer analysis of nanoparticles as a service. However, after discussion with our partners and clients, we have understood that they would also benefit from being able to do some of this analysis in-house. Wherefore, we, together with the long instruments, have developed a tabletop transmission electron microscope, which we call the MiniTEM, which you can place in your ordinary lab facility, even at the 8th floor if you would like to. So, why use electron microscopy at all? That I would say is because particle characteristics like size, shape, purity, and actually the overall morphology, and I will come back to that, of your product can often be directly correlated to efficacy and safety. Electron microscopy is a great technique to really get to know your product, to understand your design space. Like, what happens if I change my formulation and purification process in this or that way? So for all of you that are working with, for instance, viruses and proteins, you know that the process to get your product is quite complex, and that there are a lot of parameters and steps to keep track on, like aggregation due to unsuitable or up or downstream conditions, the trade-off during purification to get a pure product but not losing too much material, and of course, to make sure that your particles have the right morphology and are intact also over time. And I would now like to come back to the overall sample morphology. For years now, we have been working together with clients within the vaccine and gene therapy field. And independently what kind of particles they are working with, it always comes down to find a way to characterize the sample, the whole sample, a way to understand the overall sample morphology. What kind of structures are in the sample and what's the relative level of each structure? The tricky part here is to find a robust method to classify the structures that you find in your sample in a way so that you can compare the results from one batch to another. And when you can correlate a specific structure or even a specific structural footprint, as there is often more than one structure within your sample, to the wanted biological effect, then you have created a very powerful tool. To make it a little bit more clear, I would like to show you an example of what we did together with a client just recently. The sample analyzed in this study contained a couple of different structures. which we could classify by looking at their size and shape. The most frequent structure was quite small and almost spherical. This was also the particles responsible for the wanted biological effect. There were also some larger structures with different shapes, but these seemed not to affect the efficacy nor the safety, so these structures were acceptable as long as they were not too many. Then there were also some elongated structures with similar size as the small spherical ones, but these were more flattened. And these flattened particles seemed to actually decrease the wanted biological effect, meaning that this was a structure that the client really wanted to monitor. So here we were able to classify the particles and set acceptance criteria for two of the most important structures, giving the product a structural footprint of an accepted as well as a non-accepted product. Sometimes this is easier than others. Here, for instance, we have examples of 3 different adeno-associated viruses, the AAV particle. We know that it is the nice spherical intact AAV particle that gives the wanted biological effect. In the image to the left, you can see nice looking AAV particles. The background is quite smooth, meaning that there is not so much debris. We can, however, see one unwanted structure, which is the proteasome. So maybe here you would like to add one more purification step. In the middle, you have an AAV sample with much higher background debris. You can find the proteasome also here, as well as large aggregates around 500 nanometer. And on the right-hand side, you have a really nice looking AAV sample. However, we can here see structural conformations of what we call the duplets and the triplets. You can, of course, argue that this might be due to sample preparation using the negative stain technique, but we could also see these structures in cryo-electron microscopy and can therefore expect these structures to be present also in solution. However, sometimes you're working with more structurally complex samples, like for instance, the influenza vaccine or influenza particle. Here you often have more structures which need to be classified and correlated to their biological effect. In these samples, you often find, for instance, spherical particles, elongated particles, broken particles, and often it's those elongated particles that are broken in the end. And of course, also background debris. And you can make this list as long as you want or as long as you need. And sometimes you need to go even deeper into the structure to get a good classification, looking into the finer structural details. Here to the left, you have a nice looking influenza sample with nicely decorated particles where you can see the spikes on the surface. However, to the right, the same sample have actually passed through one additional purification step, which seems to have shaved off the most of the surface proteins. Wherefore, most of the particles looks naked. And you can find spikes as free entities in the background. So while the product to the left has good biological activity, after one more purification step, it lost most of it. So for influenza and lentiviruses, it could be a good idea to monitor the presence of surface proteins. For adenoviruses, you might need to analyze if the fiber in the 5-fold axis is still there. And for rotaviruses, you might want to make sure that it still has all 3 protein capsids. And here you can have different preferences depending on what sample you are looking at. So there are a lot of parameters that can influence the quality of the product and which can be monitored using electron microscopy and image analysis. Therefore, I will now give the word to Martin for him to tell us more about the MiniTEM system and how it can be helpful with all the automation that has been built into the system.
Thank you, Josefina, for this introduction and explaining the setting in which electron microscopy can provide insights in biopharmaceutical development. We're now going to talk about MiniTEM and the use of this system to answer the questions we have discussed previously. But first, some basics. The MiniTEM is a highly automated transmission electron microscope that provides a spatial resolution of about 1 nanometer. It's sufficient for most applications that we've talked about so far. It has all the tools available for easy exploration of samples, as well as unsupervised screening and sample analysis. Because of the automation, the instrument can be operated on a generalist level, which makes it accessible for more personnel. The instrument can be placed in a standard laboratory setting with no extra requirements of mechanical or electromagnetic interferences, which is usually the case when acquiring a full-sized electron microscope. Installation and commissioning takes a few days. Now I'm going to guide you through the automated procedures in which the system works in order to collect and provide meaningful and accurate data of a sample. And we're going to discuss how this data and output can be representative with the sample and ultimately the product being analyzed. On the slide, you see a representation of a standard 3.1 millimeter grid, which is used to place the sample upon and subsequently mounted into the instrument. To investigate sampling variability and preparation variability, it's a good idea to investigate different areas on the prepared sample grid. 5 or 9 areas are usually enough to investigate variability or trends throughout the grid. You can tell the software to visit pre-configured waypoints with a certain pattern, or you can manually, based on a low magnification assessment, tell the software which waypoints to investigate. The system then screens these areas automatically using the inbuilt operational automations. Each waypoint will be visited and each area will be imaged in a structured way. This would take a few hours if it's done manually, which for an experienced expert would not be an attractive task. So the system moves from area to area and collects images at each area based on user configuration. Each image is processed and particulate matter such as the primary analytes, but also undefined structures of interest are registered and accounted for. So this is done for hundreds of images with tens of thousands of particles available for statistical analysis. For biological samples with its diversity and complexity, the automated operation takes about an hour. Easier samples takes, of course, shorter time to process until convergence of statistical parameter estimations. Though first, and it's worth mentioning, it is a challenge to understand and group conformations of structural expressions in an objective manner. What is the primary analyte and what other objects are of interest and what would be of interest to track? From the field of psychology, we know that our own mind starts to create mental prototypes very early based on small amounts of data. The risk with this approach is that it's possible to reinforce the already established prototypes when we are presented with new confirming data, and we also risk to neglect non-confirming new data as non-important instead of updating our own set of prototypes. Many of us have heard about machine learning and artificial intelligences for image analysis. By using supervised machine learning, we can quickly test our own imagination, but there is also risk to impose our own prototypes to the algorithms that will then only mirror and confirm our own assumptions. The other way around is to create a general explainable rules for perception, which we can apply to our data, and we can from this point determine if the rules give us meaningful insights, knowledge, and ways to test hypotheses. We need both approaches in order in our efforts to push our understanding further of our products. For instance, when it comes to pleomorphic particles such as lentiviruses or influenza viruses to the right, the morphological variability is much higher than when it comes to rigid particles such as adenoviruses to the left. In both cases, it's challenged to decide which conformations that are representative and can act as prototypes for our purposes of describing the sample. And most importantly, as Josefina pointed out, which conformations can be linked to biologic effect, may it be efficacy or safety. If we apply simple rules for determining the resemblance of different objects with each other, it gives us a tool to understand the morphology of a sample represented by hundreds of images with minimum amount of bias. The result of such an algorithm can present and suggest prototypes of similar objects. In this slide, you see clouds of identified objects that resemble each other. On the left side, we see round, intact influenza particles, and on the right, slightly elongated particles. Continuing with the discovery, smaller, partially affected and ruptured particles, as seen on the left, and smaller debris of particles to the right can be identified. When we have these insights, it's possible to design automated screening tools which search and quantify these conformations in a time-optimized way. As an end result, the composition of the sample can be quantified in relative frequencies between the found conformations. An application of usage that Josefina presented previously is the possibility to assess particle integrity throughout the production process. Biological material are delicate, and all processing steps involve shear stresses to the product. I'm going to go through two situations, AAV particle integrity and adenovirus particle integrity. Now, in this slide, the visual representation of different conformations of the particles are exemplified. For the AAV particle, we see from the left to right, first what is believed to be an intact particle. The next show heavy internal staining, possibly caused by a compromised structure. Also, special observations of doublet and triplet formations can sometimes be expressed. The reason for this is not confirmed, but may be due to unoptimal buffer or temporal change of conditions causing disassembly or consequent unoptimal reassembly. The adenoviruses show a gradual breakdown, and the cause of this will be discussed a little bit later. Now, the automated workflow is the same as described previously, with screening over multiple sampling points over the sample grid, multiple images and particle detections and classifications, giving a condensed metric of the ratio of intact particles compared with all particles detected. Since the preparation procedure of samples can affect the particle integrity, both stain selection and stain quality of the grid areas are critical factors for accurate viral particle integrity measurements in transmission electron microscopy. 3 images acquired with MiniTEM show representative grid areas with different stain quality of AAV samples. On the left, B, we see a grid area with poor stain quality where the stain has been dried out. A dried area is whiter compared to the other areas, and the particles lack the typical stain halo that is observable during optimal staining conditions. When we scan for viral particle integrity status in these areas, we see a clear correlation between stain quality and particle integrity, which is an unwanted dependency. However, over a certain threshold of stain quality, the results stabilize and a comparable measurement between samples is obtainable. Adenoviruses particles are more sensitive to the effects of staining agents and compared to AAVs. Here we have 6 different samples showing the same degradation of particle integrity when the staining environment surrounding the particles is poor. The top left image shows a completely dried area, which is also represented in yellow dots, B, in the graph. The correlation between the sample preparation and integrity measurements is high with a high value interference, as in the case with the next image C and also possibly D. Over the stain quality measurement of 1, the integrity measurements show low correlation of the preparation procedure, and the interference is also low, which makes it a good observable, as in the case of AAVs, the measurement is comparable between samples. For the AAV samples, we can distinguish between the intact form and the internally stained non-intact form, giving us a separate measurement, and also the ratio of doublet and triplet particle in the population. So we have here two samples with differentiable level of both intact particles, but also a differentiable level of doublet and triplet formations. If one would keep track of these parameters, the design space could be further detailed and the effect, if any, can be monitored. For the adenoviral samples, the preparation of samples B, C, and D are disqualified, but for the samples E and F and G shows a comparably different integrity as well as internally low variability. We have this far talked about recurring structures. Debris is a different form of residual structures which can be seen as simply just the rest in the sample, which can be registered and accounted for. The condensed statistical output can be used to quickly assess the sample composition. Deviating data can quickly be investigated during simple point and click on graphs, which will guide you to the right image for further evaluation. In this slide, we see, for instance, a deviating large registered object with particles and other biological structures present. Also, when having registered the primary analyte and other non-wanted structural appearances, we can aggregate the information even more to provide a metric of purity. On this slide, we see on the left the area ratio between debris and the primary particle, in this case adenoviruses, for two different samples. The box plot shows the necessity to evaluate the grid at more position than one because of the possible variation throughout the prepared sample. This metric can in turn be cross-correlated to the product efficacy or ultimately unwanted side effects, as well as optimizing and monitoring the performance of purification processes in production. As a comparison in time consumption of performing this analysis, we have sample preparation taking about 15 minutes. Microscopes usage takes the MiniTEM about an hour, which is automated, leaving the analyst to perform other tasks, compared with a manual operation of a few hours to collect enough images. The online image analysis takes also about an hour per sample in the MiniTEM, providing about 60,000 registered particles. This is not at all a task that is feasible to perform manually, but extrapolated data shows that it would take about 2 weeks of manual work. So now concluding, the time of analysis with MiniTEM is feasible to use in routine work. So back to you, Josefina.
Thank you, Martin. So if I try to summarize here, by having MiniTEM at your site, I hope that we have been able to show you that you will be able to do detailed characterization of your products in early development, which can of course then support both the formulation and production. And by identifying critical and measurable parameters of your product in early development and by using them throughout the whole process, you will be able to develop a robust production and high quality products. And of course, we would like to be there and help you. So depending on your needs, you can use our services where we take care of the whole process. Or if you already have an electron microscope standing in your basement, you can use our image analysis software WAS. And would you like to do this in your own lab facility? MiniTEM is the way to go. So thank you very much for listening, and hopefully we have time here, yes we do, for questions. And if you don't want to put your questions in this forum, please do not hesitate to contact us directly. So Freddie, I think I will give the word to you from here. Thank you very much.
Thank you very much, both. So just a quick reminder for the audience members, in order to ask questions, you can send them in by the questions widget. So all you need to do is just type them into the box at the top left-hand corner of your screen and click on Submit. So guys, let's have a look at the questions which have already been submitted so far. We've got one here for you. So someone says, how many MiniTEM images do you take per sample?
Yeah, so it depends a little bit on the variability of the sample itself, but if we would, for instance, need to, and of course the preparation variability, but as I mentioned, when we had 5 or 9 different areas, then possibly we also take about 25 images per such area, so we would end up in roughly about 200 images per sample that is screened, and this is usually well enough to create stable statistics, but this should absolutely be time optimized from sample to sample.
Very much for that, Martin. Let's see another question here for you guys. Someone says, is the software 21 CFR Part 11 compliant?
We are right now. It is not, but we're working on that, and we can also connect the MiniTEM software with the VAS software to handle the traceability and management of the data. So and the VAS software is GMP compliant.
Thank you very much. We've got another question here submitted. I think this person may be referring to your previous answer there, Martin. They say, have you tried to validate this method?
We are, as a company, we have validated a lot of electron microscopy assays, and so we have experience of doing that. Of course, this has to be done every time for each type of product, of course. But we have the experience of doing this for both for instance for liposomal products and AAV products.
Yeah, and I can there add
that
what we have done before, I mean, then you have to make sure that you validate the method for a specific product, but also for the specific intended of use of those analysis results. So it might be that even though you have validated a method for one intended use, you need to do it in another way for another client if they are not using the results in a similar way.
Great, thank you both. Another question here for you guys. Someone says, good afternoon and thank you very much for the presentation. What is the percentage of false positives, brackets debris degradation, in a more general use of the term?
I didn't really understand the question.
We can always move on if you like.
Yeah, let's move on and we can come back to that question. Absolutely. Yeah.
Absolutely. So let's see. We've got one here for you guys. Someone says, can you do particle size distribution for a specific type of particle, e.g. only the spherical ones with good surface morphology?
Yes, of course. when we do a detection and registration of the particles, of course the sphericity or the different types of morphological measurements, such as the circularity, can be measured, and we can just have a look at these ones separately, and also do a texture analysis on them in order to pinpoint just a subset of all the particles. Yeah.
Great, thank you for that. Let's see here, another question here for you. So, someone says, have you tested any stability samples at multiple temperatures?
Yes, we have. For quite so many different particles, actually, we have done a lot of stability tests, both, stored at different temperatures, but then also over time. So, yes, the answer is yes.
Thank you for that. Another question, someone says, how many samples can it analyze per day, and what is the throughput?
I would say that you normally analyze, let's say, 8 to 10 samples a day.
Perfect. Another question, someone says, how difficult is it to create an analysis chain?
So basically, when we, it's a little bit dependent, of course, of the sample being analyzed itself. Usually it takes, maybe if we have a completely new product, getting the first results out, it's a little bit of an iterative process, but it may take about an hour to start with, and we see what we get and what is left, and this is also then we get the experience of the robustness of the workflow itself, and see what is left out, etc. So maybe to start up with like an hour, and then we can go from there, optimizing this. We've also seen that some typical types of samples, when it, for instance, comes to AAVs or MVM viruses or polio viruses, we have used quite the same starting point, and then we optimize from there. So it's together with the experience that we have, it's quite fast.
Great, thank you. Next up, someone says, how many virus types are you able to detect and classify in a sample?
As many as there are. As long as the viruses are sort of so that you can differentiate between those, it shouldn't be any problem. Of course, if you have particles like, for instance, the adeno-associated virus and polioviruses, which both are very smooth, they do not have any real structural features that you can differentiate between them on the surface, and they have the same size, then of course it could be difficult. But as long as they differ either in texture of the surface or in size or in shape, it shouldn't be any problem.
Thank you for that, Josefina. Let's see, up next, someone says, how robust is the analysis, and do you need to make a new script for each new sample?
Well, the whole idea is not to make a new script for each sample, but then to make a stable script that works for the product, and then we can use exactly the same standardized way of analysis in order to have comparable results from different samples for that product. So the basic idea is not to optimize for each sample that is put into the system.
Thank you for that. Up next, someone says, how complicated is it to prepare the sample for MiniTEM measurements?
The negative stain technique, which we use for the MiniTEM, is very straightforward. I mean, it's an easy technique, and the sample preparation just takes like 5 minutes. But it could be sample dependent. So if you have a lot of sugar, like sucrose in your sample and so on, and the spreading of the sample could be uneven, and that makes, of course, the imaging session a little bit harder. But then, of course, you can compensate by having more wave points, as Martin talked about, to say that maybe you take images as 10 different wave points or 20 different wave points instead to get the good statistical baseline.
Thank you very much for that. We've got time for a few more questions. Up next, someone says, what sample size is needed?
Normally, not more than 3 microliter is applied on the support. And then, of course, we would like to have, let's say, particle concentration of 10 up to 6 log, but I would say otherwise the volume is extremely small.
That. Let's see, up next, another question here. Someone says, would I be able to run the MiniTEM manually as well?
Yes, absolutely. The MiniTEM has the possibility so you can run it just point and click and move around and look at the sample just like using Google Maps or something like that. And since we also have the operational automation at hand, things like alignment of the system and autofocusing and these things are handled automatically, so you don't have to worry about that. But so basically, it's like working with Google Maps or something like that when doing a manual investigation. Yeah.
Thank you, Martin. We've got time for about one more question. Someone says, do you have GMP compliance / Part 11?
Right now, we do not. We have a good quality system that we work according to, and that's for GMP. And right now, we are building our own lab facility, which will be GMP compliant during next year. I really hope so.
Great, thank you very much. That's about all the time we've got. Don't worry if your question wasn't answered during this session. Josefina and Martin can get back to you personally at a different time. Guys, is there anything you'd like to add before we conclude?
No, not more than to thank the audience for listening. And of course, if it's anything, just come back to us and we will try to answer the questions as good as we can. And thank you, Freddie, for hosting.
Excellent. So that just leaves me to thank Josefina and Martin for always a great presentation, and to thank Veranova for sponsoring the session. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. Once this webinar ends, today's survey will appear in its place, and we would really appreciate it if you answer the questions in there. Also, if you click on that green resource list widget, you'll find a couple of relevant links from our speakers. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned, and follow us on Twitter @BRWebinars for daily updates, and join our LinkedIn group Business Review Webinars. Thank you once again, and I hope you have a nice day.